Hi everyone, my name is Ellie, and I spent my summer developing tools to analyze the structure of membrane proteins. All cells and organelles are surrounded by a lipid bilayer membrane composed of hydrophilic head groups on the outside and hydrophobic tail groups on the inside. While some small nonpolar substances and gases can freely diffuse through this membrane, larger polar molecules like sugars, including glucose, or ions like potassium, need the help of membrane proteins to travel through to the other side. These proteins actually interact with these ions, so understanding their structure gives us great insight onto how this interaction actually occurs. This structure can be anything from the primary amino acid chain to the quaternary interaction with other proteins. Understanding this structure gives us a look into how the protein actually functions. Not all membrane proteins function as transport proteins. Some can be receptors or help with cell-to-cell signaling, but there is a really large class of membrane transport proteins. Together, understanding the function and the role gives us deeper insight onto understanding the structure and the function gives us deeper insight into the role in the organism. This role can be anything from abiotic and biotic stress resistance to cellular communication and cell-to-cell signaling. While the role and the function can be easy to study using gene knockout, the structure is a little bit different. A common way to study any protein, not just membrane proteins, is to look at the purified form. But with membrane proteins, if we remove them from that lipid bilayer I mentioned, they start to unfold and lose their structure, causing a bottleneck in structural studies. It's really important, though, for us to understand the structure, especially in plants. As an example, 25% of the Arabidopsis genome, that's 1/4 of the entire genome, codes for membrane proteins, so they have to be pretty important. But less than 5% have been analyzed for their structure, and that's about 5 to 10 proteins. That's why I wanted to spend my summer developing tools to make this analysis a little bit easier and more accessible to more people. An example of a newly found plant membrane protein is called OSCA, which is a hyperosmolality sensing calcium transport ion. And so it helps calcium ions move to the other side of the membrane. So far, what is known about OSCA is that in Arabidopsis, when the mutant is treated with sorbitol, which mimics drought conditions, it plays a role in root growth, as well as stomata opening and closing, indicating that it plays a part in drought tolerance. For my study, though, I wanted to look at the rice homologue of OSCA. Rice is a little bit more agriculturally relevant than Arabidopsis, and the function, structure, and role is currently unknown, giving me a little bit more leeway in my angle of study. Additionally, the lab I've been working in already had a stock of the purified protein in a detergent. It's important to keep membrane proteins in a detergent solution because that mimics the lipid environment and helps it keep its structure. So the first step of my structural analysis was to remove the purified protein from the detergents and to put it into what's called a liposome. And a liposome is just a sphere of a lipid bilayer membrane that I actually made in the lab. And so we put the purified protein with the detergents into the liposome, and then I removed all of the detergents from the solution with what's called a biobead. And that leaves us with what's called a proteoliposome. And that's just a sphere of an empty lipid bilayer membrane with just my protein in it that mimics a cell membrane with just my protein, allowing us to study the protein in as close to cell-like environment as possible. And this Western blot here shows that I successfully reconstituted my protein into the proteoliposome. This band right here is just the purified protein, and then I have the band of the protein in the proteoliposome, and since they're about the same weight of the predicted OSCA weight, I can confirm that I did reconstitute my protein as desired. But before I could jump straight into a structural analysis, it was important to confirm functionality of my protein. And if it had kept its ability to function to move ions across a membrane, then it likely didn't lose its structural integrity during the reconstitution process. I decided to use an electrophysiological approach to see if my protein was still functional, and this involved reconstituting the proteoliposome into an artificial lipid bilayer. I then was able to study the movement of ions across the protein, and the way we see these recordings is as electrical current. And since I could see a change in current, that meant the ions were moving, and my protein had to be still functional and had been successfully reconstituted into the proteoliposome without losing its structural integrity. From there, I decided to jump into my structural analysis. I wanted to look at the stoichiometry of my protein, which is the number of individual protein molecules working together to form a functional unit. In this example I'm going to walk you through, I'm showing you a dimer, which is 2 individual protein molecules. It's important to remember it could be any number of individual molecules working together to form the functional unit. We figure this out using what's called single molecule photobleaching. And what this is the black lines here represent the 2 sides of the lipid bilayer membrane in the proteoliposome, with the protein situated across that membrane. Each individual protein is tagged with a GFP, or a green fluorescent protein, which, when excited with a laser, emits light that is picked up by a microscope. But when you shine GFP with a high-intensity laser for an extended period of time, it loses that ability to fluoresce, and we call this bleaching. So it no longer emits light. But not every GFP is going to bleach at the exact same time. So the first one will bleach, and then the second one bleaches. And we're able to pick this up as a change in light intensity over time. And the way we actually see this is a video like this. And if you watch this video, each individual spot is an individual functioning unit. So watch this spot in the yellow circle, and you'll see it loses light intensity over time. We use what's called a TIRF microscope, or a total internal reflection fluorescence microscope, which allows us to just see about 50 nanometers into the cell surface. So we were imaging my proteoliposomes, and we were able to just see the very edge of it, which gives us a high enough resolution to see individual molecules. What's really cool about this TIRF microscope specifically was that it was homemade on Cornell's campus. And the way we get results from this is the software picks up graphs like this. And if you watch the one in the middle, it'll show you the change in intensity over time. And this is just an example. So since there's 4 separate drops in intensity over time, that means there were 4 separate bleaching steps, making it a tetramer, because there's 4 individual units, each tagged with their own GFP, that each bleach separately. Not every protein is going to behave as a tetramer, though. Some will be monomers with just 1 drop in intensity, or it could be trimers with 3 drops in intensity. And the results that I gathered from my study with OSCA show that the most common drop in intensity over time was that of a dimer. And I counted, I looked probably through about 5,000 of these individual spots. We have to exclude a lot of that data for various reasons. But I had over 881 dimers, and that's enough data for us to make the preliminary conclusion over 2 days that the preferred conformation of OSCA in a membrane is a dimer. And that's a pretty big conclusion. But I drew that conclusion for an in vitro analysis, so not in a native cell condition. So the next step in my study was to move into an in vivo analysis to confirm the results that I had already drawn. And doing an in vivo analysis makes it more physiologically relevant because we're looking at a protein that was actually made in the cell and never removed from the lipid environment, so there was no detergent manipulation to mess up its structure along the way. At the same time, we wanted to overcome some of the drawbacks of eGFP, which is what we tagged our proteins with to begin with. And one of those being that it's less bright. It's not the most bright monomeric, or it's not the most bright fluorescent protein known. And that means we could have some monomers in our distribution because not every GFP was fluorescing to begin with. GFP also has the tendency to dimerize, meaning that we could have an extra bleaching step in our results because an extra GFP had been tagged in the complex instead of an extra OSCA molecule tagged in the complex, and that can skew our results. To overcome this, I decided to use mNeonGreen, which is a new fluorescent protein which is extremely bright and completely monomeric. So it's less likely to dimerize and skew our results with an extra bleaching step. In order to study mNeonGreen, we had to first create backbone vectors that could put mNeonGreen in both plant tissue as well as xenopus oocytes, which are frog eggs. We used the user cloning technique, which allows directionality. So the backbone vectors were with both a C-terminus and an N-terminus to the protein, which helps with localization studies. What's also cool about the user cloning technique is any protein of interest can be cloned into this casket, making it a good resource for the community. And so far, I have visualized the empty backbone with mNeonGreen in plant tissues. It was important to first visualize the backbone before visualizing it with a tagged protein, because we needed to make sure that a variety of microscopy techniques could pick up mNeonGreen and that it could be visualized in plant tissue. With confocal microscopy, we looked at it in tobacco cells, and you can see it clearly does fluoresce, and it can be visualized using confocal, as well as it's a cytoplasmic tagging protein, so it doesn't really bind to any specific organelle. It even goes into the nucleus, which you can see here. I also visualized mNeonGreen with an epifluorescence and a turf microscope, and I used the same microscope that I used for my photobleaching analysis. And it was really important to visualize with the turf scope because we want to use mNeonGreen for further photobleaching just in vivo. And if the mNeonGreen can't be picked up by the turf scope, then we wouldn't be able to do the photobleaching. And knowing that mNeonGreen can be picked up, we can use it to validate the studies that I did this summer. So this summer, I successfully evaluated the stoichiometry of the plant membrane protein OSCA, and using mNeonGreen in vivo will validate these results. I also created backbone vectors to start off this in vivo analysis that will be continued after I leave to confirm the structure that I already found, as well as continue with other structural studies like topology, which is the orientation across the membrane. And at this time, I would like to thank Miguel Pineros for hosting me this summer, as well as Dr. Krishnan, who is a great mentor, Julia Miller and Allison Colchillo, who were wonderful helps in the lab, as well as Alex Van Skyle for his help with the microscope, and then all of our funding sources, the USDA, NSF, Cornell, and BTI. And I can take any questions at this time. Yes?
So I have a question. This protein is always transporting the calcium, right?
Yes.
Do you have any hypothesis how to regulate food then?
So that study was actually done not by our lab. There's only one paper published about OSCA as of right now, and it's very just seeing what it does. So I don't think that lab has gone into the details of how to use it to control these problems, and so that's like future direction, is to use it to actually control what happens with it. Anything else? All right. Thank you, guys.